Hello and greetings to everyone joining us from different parts of the world. Myself, Dr. Deepak Parchwani, serving as a coordinator of the APFCB webcast and e-learning program. On behalf of APFCB, I extend a warm welcome to all our participants. This program was initiated to foster knowledge sharing and professional development across our global community. Through this platform, we aim to bring expert insight, latest advancement, and meaningful discussion that contribute to the growth of laboratory medicine. In this initiative, Quiddle Ortho is serving as our knowledge partner, and we are thankful to them. To guide us today's session further, it is my pleasure to introduce our esteemed moderator, Dr. Pradeep Kumar Dabla. Dr. Dabla is a distinguished expert in this field and has been actively contributing to laboratory medicine through his research, leadership, and educational initiatives. With that, now I invite Dr. Dabla to take over and lead us through today's session. Thank you. Dr. Dabla, please.
Thank you, Dr. Deepak. Thanks for the nice introduction and initiating this important session so very well. So I may take the opportunity now to introduce our next speaker. The speaker one, Dr. Tony Batrick. So he don't need any introduction. Dr. Tony Batrick is the CEO of the Royal College of Pathologists of Australasia, Quality Assurance Programs that we call as RCPA-QAP, and holds the multiple academic positions including adjunct professor at the Griffith University, honorary associate professor at ANU and Bond University, and also the visiting fellow at Macquarie University. He's also serving as the president of the Asia Pacific Federation of Clinical Biochemistry and Laboratory Medicine, and also as a member of the executive board of the International Federation of Clinical Chemistry, and is also the chief examiner for the Faculty of Science at the Royal College of Pathologists of Australasia. So I welcome Dr. Tony Batrick to present his talk under the title of Transitioning to ISO 15189-2022, Key Changes and Implementation Strategies. Welcome, Dr. Tony.
Thank you very much, and thank you for the invitation. I'm pleased to be here. My voice is a little bit raspy, so please forgive me if I'm a little bit hard to understand. So what I'm going to talk about is the new ISO 15189 standard, the 2022 version. I'm going to talk at a sort of a relatively high level about why the change. What's the philosophy behind ISO? What are the most significant changes? What issues have arisen in implementations so far? And then I'll talk about some specific differences where they're sort of major. I think what's happened with this standard, this ISO standard, and a whole raft of other ISO standards, 17025, 17043, 14,000, and many other standards, is that they're moving away from a compliance approach where they write down a clause and you basically have to follow it, to an approach where it's focusing on risk. So you'll hear risk all the time, and in fact the theme of the second talk is really about risk management. And what follows from that also is, well, what's the risk? So the risk is really about patients and about staff. And that's related to what can go wrong, and that's about competence and equipment and QC and EQA. But it's all about risk. And the reason why ISO and many other organisations, look, standards are really quite slow in making this change. Insurance companies made this sort of change, 20 years ago. And that's because it's really hard to write standards that cover every situation. If you look at medical laboratories, there's big ones, there's little ones, there's ones that are associated with hospitals, ones that are associated with point of care devices, ones that are associated with community practice. So how can you write a standard, one standard that covers every situation that might arise in those situations. So the best way to do it is to look at risk. So what are the major risks in those different situations, and to whom? And then modify the current standards, the 2012 version, so that it covers the same areas, but it puts more onus on the laboratories to identify what the risks are. So keep thinking about risk, and keep thinking about who's at risk, and then look at this standard in terms of how does it address those two situations. So it's worthwhile going back, I think, to the very start. Why are there ISO standards? And remember, ISO means the International Standards Organization, but it also means ISO means in an isosceles triangle equal. So the idea is that standards, if you follow a standard, no matter where you are, you're at the same level. You're doing the same thing. So ISO standards mean it's the same everywhere, no matter where you're operating or where you're building stuff or whatever. ISO means everybody is doing the same thing to the same standard. So the concept of ISO standards, when they were developed, was that if you're accredited once, you're accepted everywhere. So that's important. The previous standards, the ISO particularly 15189 has been built up over many years based on 17025 initially, and then specialised to medical laboratories. But it's comprehensive in the nature of its requirements, both the management requirements and the technical requirements. Many of the technical requirements coming from 17025, many of the management requirements coming from 9000. The standards all fit together. They're ISO as well. But it's the only standard that's applicable to medical laboratories. And it's also recognised by most governments around the world in one form or another. In Australia, every laboratory, medical laboratory, has to be accredited to 15189. And I know in India, there's a significant number of laboratories that are also accredited to 15189. There are continuous updates of the requirements. It's been a long time. This time it's been 10 years. Normally ISO standards are updated every 5 years. And the purpose of that is to make sure that they reflect what's changing in laboratories in our case. And it's an international non-government organisation, and that also ensures that it's impartial. So what it's doing by bringing a group of experts, a lot of experts from all around the world to write these standards, is that they're independent, that they really reflect best practice in laboratories, and that's why they've been endorsed by most governments. The differences between the earlier versions, 2007 and 2012, and I say ISO standards are normally updated every 5 years. Really, the requirements of laboratory management systems, they've developed so much between 2007 and 2012, and automated results reporting. So that was a major change between 2007 and 2012. And really, this was because at that time, a decade or so ago, there was a rapid introduction of automated systems into most laboratories, and really a revolution in terms of computer software in medical laboratories. In 2022, again, we can see what's happening out there. Point of care has now become a significant player in both hospitals and in community practice. Therefore, that was incorporated into this version of 15189. So the standard 22870, which was the old ISO point of care standard, is now part of 15189, because most laboratories are responsible for some sort of point of care network, particularly if they're associated with a hospital. There's also, when you look at what's happening in laboratories over the last 10 years or so, there are now laboratories that really are doing tens of thousands of samples a day. So again, you have to develop systems that provide safety to patients and staff when they're doing that number of samples. So, it's important to adequately identify patients and their results, and also get the right results on them. So that's a reason why there's been some of these changes put in place. And I say, when you get to the sort of scale of operations that we've got now, it's very hard to compare that sort of laboratory, as I said earlier, with a small laboratory. You can't say, well, you've got to have 3 of these and 5 of those. You really need to move to this sort of general risk-based approach. How do you identify risk, and how do you mitigate against it, and how do you demonstrate that you've done that? Not all requirements were always relevant for everything, but specific requirements need to be defined for each site so that technical competence could be established on an objective basis. And we have to move away from that more to a risk base rather than this compliance base, as I said. And ISO 17025 was also reviewed, and it is the basis of 15189. So that's another reason why 15189.22 had to be updated, taking into account the changes that had been gone into 17025, which was really about risk and the management requirements. They've flipped things around in terms of the way it used to be laid out. The management requirements came first and then the technical. They've changed that within 15189 just to be more consistent with other standards. And there's also an incorporation of some of the components of 17043, which is the EQA proficiency testing standard. So again, putting all those things into one place, it means that the same requirements for laboratories are there as for the providers of EQA material. And the other thing that's happened is that these days laboratories have become truly interdisciplinary. It's hard to know whether serology samples are being measured or the immunology samples are being measured. It's usually in the chemistry department because it's all done on high volume equipment. And it's not anymore just interpreting results. It's all about IT. More and more, particularly in chemistry, we're worried about metrology, but there are certainly interesting legal requirements now on us in terms of our responsibility and obligations to patient and quality assurance. Also, what's in this version of ISO 15189, because metrology has become so important in terms of traceability, a lot of concepts, a lot of new terms have been introduced from the GUM and from the vocabulary of metrology, just so that becomes part of our everyday jargon, because it's important that we now start to move outside the ordinary laboratory. So it's not just making sure that you get the same results every day. What's really important in terms of patient safety is that all laboratories get the same sort of result. So this concept of traceability is critical and will be more critical into the future. Where terms may have a legal implication, and because there's, again, concern about legal obligations that laboratories have to patients and to government, they've also been clarified. So terms like patient, complaint, consultant, impartiality, they're much more better defined than they were in the past because they have there are legal implications. Again, external quality assessment is now more conceptual, and it's consistent with 17043. So all these things are one, so that all the standards seem to talk about the same thing, and we're looking more and more at issues such as traceability. And of course, commutability, linked to the ability to measure traceability. Commutability is a key concept for EQA providers, and more therefore for laboratories too. So some of the new requirements, this sort of got my slides seem got a bit mixed up. So in terms of the sort of new requirements, the drafting team, when they put this together, sought to limit the introduction of new requirements and to avoid overwhelming laboratories while still addressing the critical areas for quality and competence. Okay, so that was when the drafting committee, that was part of their marching orders. Don't make this too complex. Try and simplify it. We want to introduce new terms that are relevant, but we also want to try and keep this as simple as possible. Team focused on refining the language, so the language is much more readable, and the structure of the standard to be consistent with other standards, as I said, but also easier to interpret. And the drafting team acknowledged several concerns raised during the review process, and they addressed them. Some stakeholders, particularly from national accreditation bodies, perceived that any differences in text would signal new requirements. suggesting intentions which were not no objective of the drafting team. So again, if you change standards too much, there's the implication that there's something new there, but really the changes relate to, apart from those new topics I've mentioned before, changes to the standard really reflect clarification rather than confusion. And certainly risk-based thinking was key. So one of the concerns was, in fact, this issue about introducing risk, and that you somehow had to be very, very specific in the way that you did your risk assessments. You'll hear more about that next, how you actually do a risk assessment. And in fact, that's the greatest change, I think, with this introduction of this new standard. It's something we've not done before. It's easy not to do well, and it's easy to be misled that you've done a good job on your risk assessment when in fact you haven't. So that's the key, I think, to a successful implementation. People will say, if they have to have done a risk assessment, they'll say, well, I don't think there's a risk there. They haven't documented, or they haven't gone through a structured approach to assessing whether or not there's a true risk there. That's the biggest problem. But the standard does provide some practical examples to illustrate this thinking, to assist. But again, people will often take the easy way out and ignore that. So a whole lot of new terms that are in there that weren't there before. These are the metrological terms: commutability, complaint, consultant. Again, they're better defined so that they're more legal. The legality is much more clearer. Management system, measurement, MU, etc. They're now well defined within the standard. And I keep saying risk analysis. It's all about risk. It's a big change for laboratories. We're used to just following rules. Now we have to think about what's the implication of what we do to patients and to other staff. And that risk involves, thinking about every situation where there could be a failure, and how is the laboratory going to deal with that? Failures from instruments breaking down, problems with calibration, to loss of power. How can you still provide the service that you need to a hospital, or particularly to key parts of a hospital, if in fact there's been a power failure or a failure of an instrument within a laboratory? They're the sorts of risks you have to think about as well. And as I've said before, because it's now risk-based, and not compliance-based, it's certainly less prescriptive than it was before, because you can sometimes follow things blindly and not still realise why you're doing it. But what the risk-based approach does is that it invites laboratories to put into place, identify risks, put into place ways to mitigate that risk. But the basis, when your accreditors come round to accredit you against the standard, you must justify why did you do what you did. That's what's different. And the first time you go through the process, it's a bit scary. Just some things in passing. There's no longer a requirement for a quality manual. That's one of the, when we say it's less prescriptive than it used to be. You don't need a quality manual anymore, as long as you can justify that you've got a process in place that everybody follows, and it's updated regularly. There's no longer a requirement for a quality manager, and that's philosophical in that what had happened, I think, in many laboratories was it was felt that the quality manager was the person who was responsible for everything, but in fact, that's not really true. Everybody is liable for something that might go wrong. It's hopeful that by looking at risk, everybody looking at risk, that there'll be more improvements in laboratories than there probably has been in the past. The other thing that's different is, what's all this risk about? Well, ultimately the risk is about patients, so there's a much more patient-focused, based on risk approach to the way that you manage your laboratories. And that revolves around, well, if something goes wrong, how will this affect the patient? If something goes wrong with the power in the laboratory, which patients would be impacted if you're in a hospital? How do we mitigate that risk so their results continue, where the critical results continue to be produced. So that's where the risk base is. Not before, you might have thought if the power goes in the laboratory, well, we'll all go home. Now the emphasis is, well, what impact will this have on the patients? So what's my process to make sure that I don't have any potential risk to the patient? So I'll keep reiterating this, but it's about risk, and it's about risk to the patient. The validity of any sort of risk-based approach that you undertake has to be pertinent to the clinical decision making. So again, instrument failure, what impact does it have on the patient? Potentially, what impact might it have if an incorrect result went out to a clinician? How can we mitigate against that? So, for instance, whenever external quality control fails, you need to do a risk analysis. So that just in terms of, well, it's failed, have results gone out, what's the potential impact on patients? Could this have make a significant difference to a diagnosis or monitoring? It's not about QC failure anymore. It's about the outcome of that and the impact on a patient. And therefore, and not surprisingly, complaints usually come from clinicians, occasionally from patients, but the clinicians are complaining on behalf of the patient. So that's the key thing. Also, again, relating to risk, we all know that right now cybersecurity is critical, and that relates to confidentiality of patient results and data protection. Data in the laboratory is a target because there's so much information there that could be, potentially very valuable to particular patients and governments. So that's where the high risk is. But the laboratory is ultimately responsible for the laboratory management system, and therefore any patient outcomes that might be impacted by a loss of that data. Therefore, it's critical that laboratories, when they implement or change their system, that they take that into account. The details for sort of risk requirements were detailed at the level of key requirements, but the new version does not specify how to do a risk analysis. That's going to be discussed next. There's no formula for doing risk analysis. That's up to you which one you use, what table you use, what matrix you use, what process you use, as long as it's robust and you can defend what you've done. And remember, the whole reason that this has come about is because potentially you could be sued or charged if something goes wrong. It's in your best interest to do this properly. So, but the approach really is in terms of risk, what can go wrong, how often can it go wrong, what are the consequences, and therefore there's a much greater focus on process. So what can go wrong in the process, how often might that happen, what's the impact on patients? So the key issues in transition that have been identified, certainly in Australia, is who can authorise personnel within a laboratory to perform specific tasks. Really, one of the high risks we've got in laboratories is people. The equipment's pretty good. Our systems are usually pretty good. Often people aren't as competent as they should be, and it may well be that you find that a person who isn't as competent as they should be is releasing results that they don't realise are significant to patient safety, and that's where your greatest risk is. Again, the verification and review of examination methods, again, you have to ensure that your methods are appropriate, traceable where possible, but appropriate for best patient management. That's not always, in Australia, it's been found that labs still sometimes use out-of-date methods. The evaluation and clinical significance of non-conforming work, again, a failure of QC, a failure of EQA. What's the process that you go through to ensure there's been no patient's harm? So that's been not done that well. And again, audit planning based on using a risk schedule. So laboratories have to do internal audits, but you can audit everything, but you should be auditing those processes that are most risk to patients or to laboratories. So you need to change the way you think about audits, and don't just do audits for the sake of it, do them for the sake of the patient. And use a friendly risk identification form. So a key benefit is that any sorts of a risk assessment that you do, or where you identify that there's a potential incident or a hazard or an opportunity for improvement, you need to make that easy for people to identify, because that's where you identify your risks. You need to conduct risk assessments, and you'll hear more about that. You need to ensure that there's somebody responsible for these risk assessments. It should be a number of people, not just a quality manager, because they're not that as important as they were. But the key thing here is that you need to identify potential risks to patient care in the pre-examination, examination, and post-examination process, and they then need to be mitigated. And you mitigate risk through quality improvement planning. So on the basis of an identified risk, you then go through and record decisions made and actions taken on risks and opportunities and any complaints that have been made. That's how you demonstrate that you identified a risk and done something about it. And one way you can do this is to identify risks by their status and a risk rating. So is this high risk to a patient or low risk? And clearly, depending on whether it's high or low, you can then take an action that might be more involved than others. So key differences, this concept of impartiality. We haven't had to demonstrate before that laboratories are impartial in the way that they buy reagents, buy equipment, deal with patients, deal with doctors, deal with staff. Confidentiality, I say that's critical, particularly in terms of the IT system. There's much more emphasis now on ensuring that you have these processes in place and that they're understood by everybody. Again, just generally how laboratory defines. Yes.
I'm sorry to interrupt, Dr. Tony. No, we have 5 minutes left.
Okay, that's all right. I think I'll do that. Yep. We now have to better define the organisation and management structure of a laboratory. That wasn't as important as before. The quality management now has been it's not just in the hands of one single person. It's in a whole the whole staff are responsible for these sorts of actions, effectiveness of activities, reporting to laboratory management on the performance of the system. Risk management, as I keep saying, that's been introduced. It's now a key clause and important. More specific information about personnel. This competent persons has been there before, but now they need to ensure that they're also impartial, they act ethically, and they have a process management competence of its personnel. So they need to demonstrate that they're actually ensuring that their staff, who, as I said, are the major form of potential risk, are in fact competent. Internal quality control now is much more. It's broader than it was before, allowing laboratories to have a bit more scope in what they do. You can see how much bigger it is than it was before, and similarly with EQA, it's much broader than it was before in the 2012 version, just to allow laboratories to have a little bit more scope in what they do. And again, complaint process now. And there's more detail about the management system. These are the major changes. So the key issues that have been identified, again, authorisation and personnel, periodic management. I think I've actually covered this before. Evaluation of non-conforming work, risk-based planning. It seems to be going through the same thing twice. And this is just some graphs from another ISO standard that details how you should do risk management. But again, that'll be covered in the next section. So in summary, it's a more real world standard. It reflects more what's going on. It's been updated because of the changes in laboratory practice. It's all about risk and patients and the things that can cause risk to patients. And risk is a difficult concept. It is hard for laboratories to understand how risk can be applied in the way that it needs to be applied. That's where the learning will be for all laboratories. And that's it. Thank you.
Thank you, Dr. Tony. Thank you for this wonderful talk and detailing about comparatives, about the new standards, and emphasizing onto the risk assessment, which is actually a very important part of the current standards. So if you're okay, would it be fine that if we take the questions at the end, Dr. Tony?
Certainly.
Thank you so much. So we invite our next speaker, who is going to speak under the title of Risk Management in Medical Laboratories, Practical Applications of ISO 15189-2022. Again, our next speaker is a very eminent person in laboratory medicine and attached with IFCC and EFLM, and renowned person into the field. So Professor Thomas Zima is a graduate of First Faculty of Medicine, Charles University, Prague in 1990, and is also a professor of medical chemistry and biochemistry and heading the Institute of Medical Biochemistry and Laboratory Medicine. He served as a dean during the period of 2005 to 2012, then 2014 to 2022, and was a visiting professor at the University di Milano in 2024. With research interest in oxidative stress, age, nephrology, and tumor markers, he has authors more than 550 articles and 10 books with 82 chapters. And he's also president-elect of European Federation of Laboratory Medicine for the coming 2024 till 2025. So I welcome Dr. and Professor Thomas Zima. Thank you very much.
Dear friends, thank you very much for kind invitation to this webinar focusing on the new ISO norm. Tony is describing the story about this new version, and it's looking on the slides and talk. Every slide is a talk about the risk, and I will focusing on this on this part, and the what's focusing on this version. It is emphasis on the risk management and its action to address the risk of an opportunity for improvement in all processes in the lab, and the risk is sharing between all stakeholders. The helping documents to working in the labs is also the requirements for risk management is ISO 22367 issued in 2020, where it's deeply describing the topic of the risk and knowledge is about this, maybe the new era in the lab, because in past it's only the non-conformities and about the risk is only with a few parts. Risk are discussed in many chapters. I put only the few of them. 5.6, it's risk management, but it's a sample exceptions, non-conform works, emergency preparedness planning, improvement, non-conformities and corrective action, evaluations. But I think it is a red line passing to all ISO 15189. It is a risk management and this part. In chapter 5, it's looking to the governance requirements. It's maybe the one key part. It is implemented, maintain processes to identify risk of harm to patients and opportunities to improve patient care. It is a key point of it. And new approach to risk management is identifying opportunities and improved, as I mentioned, in all area of laboratory activities together with other stakeholders. In part 7, in general part, it is very important to identify the risk to patient care in all processes for pre-examination, analytical part, post-examination processes, and do all the best to mitigate this possible risk. And at the next part of my lecture, I will showing how it's counted the risk, how it's frequent, and if it's a critical part or if it's acceptable risk. And it is based between the identifying the risk and also opportunities for the improving the patient care. In the management system requirements, it is, I think it's a main part, the identification it and acting on risk and improvement. And that is important that set the priorities, because there are we should have a long list of the risk which should be done, but we must looking for the critical part which should dramatically influencing laboratory work. And this is maybe focusing, and this during the improvement and implementing the ISO 15189, we should edit the other risk and the balancing how it's done in the last period and improving. I should focusing what is the concept of risk. The concept of risk it's have two components. One, it is a probability of occurrence of harm. It is an injury or damage to health of people, damage of property or environment. It's not only in the direct laboratory part, but also about our ecosystem. And consequence of that harm, that is how severe of harm may be. The harm is that the one person of staff should have some health problem. But it is not a critical part. But if it's a laboratory information system is broken down, it is a very serious harm in the processes. And what is important that for the risk management, it is also sharing the responsibilities between the laboratories and also the IVD manufacturers, because the IVD manufacturers is adding the information about the risk in the laboratory equipment, in the in vitro diagnostics, and this is putting together all stakeholders, sharing the risk value of the harm in the laboratories. Also, it is the regulatory authorities in the country, some other municipalities, governmental institutions, and also accreditation bodies, because accreditation bodies should looking for the system if it's effective or not effective. Also, there is maybe sharing the risk with the stakeholders, the external quality control organizers. We must putting together that this part, if we will receiving the data for equal organizers, that is okay and fitting to our system, and it's very important. Risk management, it is should plan a systematic process which go through all part through the ISO 15189 from audits, corrective action, quality control, management review, and as I read, it is a red line through the whole ISO. Now it's two slides about the terms of definitions. Risk is combination of probability, it's chance if it's expressed between yes or no, impossible or absolutely certain, of occurrence of harm, it is, as I mentioned before, injury or damage of the health of people, property, environment, and severity. And measure of this, it's very important because it's sources of potential harm. It must be balanced severity and compare together probability, harm, severity together. Risk analysis, it is information about an identification of the hazards, sources of potential harm, what should be done and estimate the risk of that harm. And it includes also the examination of the events which should be produced the hazardous situation or harm. Risk assessment, it is comprising the risk analysis, as I mentioned, and risk evaluation. It's comparing the estimated risk with the risk criteria to determine the acceptability of the risk. And as I mentioned, the risk management, it is a whole systematic processes which is done. Risk management, it is have a few elements. It's a risk management plan, analysis, evaluation of the risk, control, management review, and monitoring the risk through the internal audit system, as example in the lab. The scope of the risk management plan, there are some parts and not is the limited of these factors should be considered to the management plan. It is a quality of specifications, critical part, it is the medical decision levels and critical values, because if we have a bad critical values or reference ranges, I put the examples, differences between men and women, differences between the age. If we will putting the one range for ALP, it's very, very harmful for the children because it's different. Patient populations, measurement system and measurement of uncertainty, it's also very, very critical part, because if it's the uncertainty of our measurements is out of the ranges, it's we put false data and not be precisely fitting to our medical decision process. Performance analytical characteristics, also the pre-examination contact with the patients, preparing the patients for the phlebotomy and sampling, temperature, transport, and other parts. And clinical use of the examination result, because the different tests for screening, diagnostic, confirmatory tests, which may be putting on the different levels in the management plan. The identification of hazards, it is, I think, the critical part. It is incorrect results, misidentified results, and delayed results, because it should lead to the maybe to the death of the patients, and it is maybe the number one of the hazards to patients. When we are put to all parts, we must implement it to potential hazard situation. It is also part from the manufacturers. The quality of the IVD, receiving the IVDs to the labs, information for the manufacturer, looking for the batches, and so on, and also the service and part of the medical devices using in the labs. Hazard to the patients, if we will have the examinations of the worse results, it may be the inappropriate medical actions result to injury or death, failure of the appropriate medical actions, incorrect or delayed examination results, which may be also the very serious influence for the health of the patients, and this malfunction in the labs, it's leading to the inappropriate medical care. There is also in the part, it's not only the scale, but for, as example, immunological results, positivity or negativity tests, it's including correct, incorrect, inclusive. The hazard which are from the fault conditions, it's, as I mentioned, there is sharing the risk and hazard with the IVD manufacturers. It is the inconsistency, as example, batch to batch, not traceable calibrator, in the lab, the stability failures, failure of the hardware or software in the equipment, packaging failure. It is also the problems for the manufacturer, and when it's entrance to the lab, if we will not accept it as example. Incorrect patient information, it is a worse identification number, date of birth or age, incorrect gender, which also leads to the worse data. There are some examples of the hazardous situations in diabetic patients, falsely elevated glucose level, patient is in hypoglycemia, false normal troponin results, a patient with the chest pain, misidentified samples on intensive care units, electrolytes in the extracorporeal surgery, or electrolyte results worse in the hemodialysis patient, etc., and etc. There are some examples of the events and circumstances which should leading. One, it's incomplete requirements of the samples, it's inadequate specifications. In laboratory processes, it is a sample volume, hemolyzed sample or influence of lipidic sample, insufficient control of the changes of the laboratory procedures. Most these things, internal quality control, insufficient control materials. In sample transport, there may be the many problems in the packaging of the sample, temperature, environmental conditions, sample preparation. It's a wide spectrum of the problems. And when we putting together the hazard situation and possible harm, for the inadequate samples, low volume, new samples required, hazard situation, it's receiving incorrect results or no results or delay of results. And harm is erroneous diagnosis or delay the diagnosis of treatment, and maybe also the death in some critical laboratory parameters. Other hazard, it is no action with unacceptable quality control results, no action with the samples, no action unacceptably controlled results. There is incorrect results for the patient, and also maybe coming to death and erroneous diagnosis. If we looking for non-conformities which leading to significant risk, I separate to pre-analytical, analytical, and post-analytical part. In pre-analytical, it is e.g. Patient preparation, collection of time, incorrect container or preservatives, very important for the example for a glucose, but also for measurement of some vitamins, as example, mismatching of the specimen, incorrect transport to laboratory. Mostly the problem with the temperature and the long-term transport. Maybe the one example in the hospitals, immediately sending the blood by the press tube hose to the lab, which leading to hemolysis. In the analytical part, it is the most problem with the discrepancies in quality control results, equipment errors, which should be lead to the delay of the results, turnaround time, mismatch of the samples for start and routine process, inappropriate reference values, as I mentioned, and problem with the new reagent lots and shipment, and also calibration. It is a potential non-conformities lead to the risk. And in post-analytical phase, it is incorrect transcription, report sending to the incorrect person, to the incorrect word. There is a problem with misinterpretation of the laboratory results, mostly some improper mutation nomenclature in the genetic test, and it's very risky for the patients. How we put the risk together and in the risk management? There is a probability of the harm scale in semi-quantitative from the level 5, which is frequent, should be each day, to the unlikely, then maybe the lesser 1 year. It is a semi-quantitative scale. If we will moving to the other one, there is a harm of scale from critical, serious, significant, marginal, it depends. And this we will put together to the matrix zones, probability and severity of the harms. And there is some parts which showing that the green one, it should be acceptable risk. But in the red one, it is unacceptable risk, and we must make some actions to change the situation. We also should use the risk matrix with the 3 zones, where will be the reasonable possible zones when we need the changes of the system in the lab. And finally, here the risk reduction, the decision, it's there is in some examples in the sample collected from the work patients, severity critical, prevent, unlikely medium, sample transport delayed or late, it is the marginal maybe for severity, but in occurrence, It's looking at the prevent action monitoring, and unlikely is very low. Which risk tool analysis and techniques? We have three widely used methods to calculate and working with the risk analysis. It is a preliminary hazard analysis. It is inductive methods where it's identifying the hazard situations and causes of the harm and facilitate the system. For the analysis, it is the analysis hazard from the top event. It is a deductive methods, and I will show you the one example here. It is the deductive methods, the causes of call, asking what, where, when, why, and what we need, the knowledge of the system and the function of conditions. And there is a file 3, top event, important disorders, and partial problems. And there is one example for the list failure. Power outage, the system does not respond. Power supply interruption, incorrect operator intervention for the not response, blocking access or viruses. And there is for the failure tree analysis from the top event, it is the failure of laboratory information system to the potential problems which should be. For that, we will need the perfect knowledge for the system. The next one, the risk analysis tool, it is a failure mode and effects of analysis, which called the FIME-A. It is a technique identifying the components together. There is the answer question. It's what's happened if automated analyzer falls. What's happened if the IVDR kits not receiving the labs? And there it should be design done for the new assays examinations, for the analytical and multiple components, and application is used for the preventive errors and processes with IVD medical devices. It's will be more smoother in this processes. There is an example of FIME in pre-analytical phase. There is a function, sampling room, sampling centrifugations, potential failure, non-compliance, incorrect barcoding, possible effects of failure, delay reception, sample confusion, analysis is not possible, and so on. There is a counted the severity. We should put the scale from 0 to 10. Potential causes, unavailable sampling chair for the sample recipients. Request is not clearly marked. There is an incorrect request, a non-compliance monitor, reception area. The occurrence also is counted by the lab. It is done not by other person. It is in the lab, in lab management, put the scale to severity, put the for occurrence, and also for the detection, how it's counted together. And there is a preventive measure on this example of the female failure. There is a recommended measures. What's here? Responsibility. Who is the responsibility? Laboratory director, supervisor, a quality manager. What is implemented to be better? There is an example for in the sampling room, the training, in the incorrect sample identification. It is replacing abbreviation of test, highlighting the codes. For the 5B, which was the incorrect activity if the list, change the location in the monitors, and the other one inspection. And we have the numbers which was in the previous slides. And the laboratory services, it's about the quality for our part, which have the impact on the care of patient, but also for the environment, for our ecosystem, as I put, and we are, maybe I put if in Asia and in Asia Pacific, when I was in many time in Taipei in the last Congress in Sydney, there are the many boats I put from our Central European country, the ship where we are all together with IVD manufacturer, we need to satisfy patients, high quality processes and lab processes inside, from pre-analytical to post-analytical part. But don't forget that all quality system is about the people, with the people, and for people, and it's also including the risk of harm. Thank you for your attention.
Thank you, Dr. Thomas. Such a very interesting presentation again, and revolving around the life cycle of the sample from pre-analytical to the post-analytical phase. Of course, because it is very pertinent that with such an interesting presentations, we do come with several of the questions, but of course, the time is a limitation. So I'll start with a few questions. My first question is to Dr. Tony Babrick. It's been, and I'm also combining my query with the same as well. And the question being proposed by K. Wang Choi. And the question is that, is ISO 15189 is adequate for regulating the use of artificial intelligence in lab medicine? And now I'm adding my also one comment that with more of a distance reporting and AI image analysis, this question also, becomes very important when we are talking about the usage of AI techniques in laboratory analysis. Dr. Tony Babrick.
It's a good question, and it's a question that we're all thinking about. I mean, ISO 15189 is a very high level standard, and if you think about the basis of it is risk, and also cybersecurity. The risks from AI and really any sort of technique like that, machine learning, etc., are about really risk to patients. In that, is the system validated? Does it give consistent results? Is the algorithm? Do we understand what it's doing? Do we understand why it does what it's doing? Is it generalizable? And the other aspects of, I think, AI systems that concern us are what happens to the data? Are you sending the data out to a cloud or somewhere else to a server where the algorithm actually looks at the data and does whatever it's going to do, and then it sends you back the result? If you're sending your patient data, your critical patient data, off-site for this analysis, that's high risk to the patient. So there's an issue about confidentiality and privacy, and obviously that may be another portal whereby somebody who wanted to get in could breach. So it's cybersecurity. So at the highest levels of AI, machine learning, risk, confidentiality, cybersecurity, I think and really there's a lot of onus in there about the LIS, LIMS is the laboratory's responsibility. It needs to be aware of that, and AI really is part of the LIMS. I think there's the complexities of AI go beyond those simple concepts, but the simple concepts tell you that yes, it'll work, but you would need more advice before you start implementing any sort of AI system. You would need more advice about where's the data going. Is it potentially a risk to cybersecurity? And do I actually believe? Can I trust that this is producing? You need to validate the algorithm in your laboratory, because that's a risk.
I totally agree, because as you're progressing more toward the AI and its implementation in healthcare, which is still in its infancy stage, I must say. To the other side, that who has made that algorithms is one thing, and the second thing is that who is being using it. So that who is going to be responsible if some faults that comes with it. And as you mentioned very rightly, that cybersecurity and confidentiality always, a matter of importance when we are sharing the data, especially of the healthcare. Now, that question that comes, vice versa to Dr. Thomas, that question again comes that can AI be useful in risk management analysis within the lab medicine? Again, another question by a participant.
Yeah, thank you for this question. I think that the AI, it's a very good instrument to helping us in the different areas. Yeah, for education. And I think that we should asking and should work, where is artificial intelligence due to the machine learning knowledge, identify the risk in the lab. And it should be helpful for us to not forget for some part in our processes. But I'm looking for the artificial intelligence as an instrument to helping to us. And if the AI helping in the areas of the knowledge, education, preparation, some documents, I think that the AI should help generally. But the AI have the general knowledge from all continents. But we are in the one lab, which is in one ecosystem, which have the defined staff, define the environment, define the equipment, number of samples, and the knowledge using for AI should be implemented in the definite lab in our thing. I think that should be helpful. Yeah.
I'm sure, actually, as you mentioned very rightly, because the forum of laboratory is very important. Like, what is a need-based requirement? And that's different from lab to lab. That is different from the scope to scope, definitely. It cannot be the same implementation from small laboratory to the very large laboratory. Very rightly said, Dr. Thomas. And coming again to a next question by our participant from Asia Pacific, Alvin Yang, though I'm not able to, because the country is not specifically mentioned. So is there any value adding to the embark on, implementing the ISO, which is widely being widely advised and widely being used as compared to the CAP accreditation? So the participant is asking, is there any value addition when the lab is already CAP accredited, adding the ISO accreditation?
But both standards effectively use the same base. Oh, so am I still on? Both standards use the same basis. So they're and CAP is becoming more and more closely aligned with ISO. It means, there are costs to accreditation and costs to the visits by the assessors. I think that it probably doesn't add a lot more in terms of the quality of the system. And I guess it would depend really on what local rules you've got, as you said, depending where the country is. There sometimes in countries like Australia, we have to be ISO 15189 accredited, and in other countries it's not essential. So you should be. They're both very, very similar in terms of what they're seeking to achieve. One is more US centric, so it's been designed by the US, whereas ISO is really global. But people often go down the CAP accreditation process because of other reasons. They need the accreditation to be able to be involved with clinical trials, etc. So there are often different motivations for going for CAP accreditation.
I shortly comment. In Europe, is the ISO 115. It's a general norm for accreditation in Europe.
Yes, it is most widely accepted. ISO 15189, absolutely right. And even in India, that the many laboratories, they do have the accreditation from the CAP as well. And as Dr. Tony already mentioned, that for some special reasons, is it that might be for the clinical trials, or maybe to promote the medical tourism even. So that might be the answer. So coming to a very interesting another question to Dr. Thomas. Dr. Thomas. What is the retention time for the infectious and non-infectious molecular diagnostic samples? Because only in ISO 22 retention period mentioned for the extracted DNA and RNA. So is there any difference between the infectious and non-infectious samples for retention?
It depends generally. There is a standard for infections diseases. It is maybe the analysis very very rapid during the, I think, 1-2 days. Yeah, we have the COVID time, and the response in infections diseases, it's very rapid. For the human DNA analysis, it also depends. If it's in some preventive screening program, and if it's not the harm of the patient for the delay, it should be weeks. But if it's make a decision for the precise treatment in the oncological patients, it should be no more than a few days or 1 week. Yeah, it depends on the situations, and also the labs should doing the results as soon as possible, according the needs of the medical decisions or the treatment. Yeah, and it's maybe this, I put the 3 scales: immediately, days, up to 1 week, and later. Yeah.
And that's one of those
because,
it's a risk-based question too.
Yeah.
It's a risk-based question too. Depending on your situation, it's about what do you need in terms of, if it's an infectious disease, have you missed it? So how long do you have to keep the sample before whereas if are they repeat samples? Are they stable? So it's all about everything goes back to risk and risk to patients ultimately.
And also, that ISO is again a guidelines. So certainly they talk about the minimum standards, and of course, as the need-based requirement, these requirements can be modified. But of course, taking care of the minimum standards, that's the guideline always talk about. So thanks, Dr. Thomas and Dr. Tani. And going again to the next question, which may be the last question probably, because we still have a long list of questions. So probably we may request you to share with those questions in a Word format, and we may request you to share your opinion or suggestions on those questions, and then we will share with all the participants on group. So one very important question is that always comes when we talk about the audits. So is it compulsory for the lab staff who is planning to undergo accreditation to have the ISO 15189-2022 audit training? It means that who are the lab personnel, importantly, who requires this audit training certification?
You need to do internal audits. Yeah, sorry, sorry to jump in. You need to have the capability to do internal audits, and really you can't do those without training. That audits are difficult to do. So I think it's worthwhile having people properly trained to do audits. In terms of how many people you have trained to do those audits, I think there's a cost associated with this, both in terms of paying for the course, but also losing people while they go away and get trained. I think, again, it depends on the laboratory and their situation. They need to make that sort of decision. But I would certainly suggest that it's worthwhile having some of your staff trained independently on how to do audits. I think that's very important. How many? I think it depends on the laboratory situation.
All right. Thank you very much to both of you for your valuable time and such an highly expert advice and talk and suggestions into those pertinent, very important topics related to the ISO 15189, both the changes and the risk assessment, because all things are, based onto the same. So I'm very, very thankful to both of you, Dr. Tani, Dr. Thomas, for sharing your valuable advice and talk on this APFCB e-learning and webcast platform. And on behalf of communication publication team in APFCB, I'm very thankful to both of you. And I'm very thankful to our participants, those who stayed with us till the last and participated very wholeheartedly, and so many questions I still have to ask to you. So after thanking, I would like to advise our participants, after the ending of the session, you will receive the link to download your certificate. So please don't forget to download your certificate after the end of the session. And certainly very important is this survey that has been programmed for on the basis of the talk and on the theme of this session today. So please, participants, don't forget to complete the survey, which might take not more than 5 minutes. So thank you very much, and thank you very much to all of you. And we will inform you for the next session very soon. Thank you very much.
Thank you and good afternoon.
Thank you very much for kind invitation. Nice weekend. Bye.
All the best. Bye.
Bye-bye. Bye. Thank you.